Alright, so section 6.3, as with the other sections in Objective 1, you have some of the terms there that will be found in this section, specifically binary fission, generation time, and turbidity. And then Objective 7 has figures 6.9 and 6.12 in it, which is really the meat and potato of this section. Alright, this is figure 6.9. That goes through the steps of binary fission. And binary fission is the main way that bacteria cells are going to replicate. And in terms of replication, this is what is also referred to as growth. All right, so when you and I grow, you think about us getting taller, getting wider sometimes, unfortunately, putting on more muscle mass, that type of thing. But with bacteria and these type of cells, the cells just aren't growing to get bigger. They're growing to replicate and to reproduce. So sometimes those terms are used interchangeably in terms of, I'm going to go grow some bacteria. All right, I'm going to go start a culture, and they're going to replicate. They're going to increase in number. So you start out with a young cell, and this guy here is the young cell. All right, it's small, and then it's going to replicate its chromosome. And in this case, you do see that the cell elongates some, so it is growing in terms of getting bigger, but it's just simply expanding itself so that then after X amount of time, there's going to be a protein band that forms in the center, and that's going to be fusing or cutting in half the cytoplasm and fusing the cell membrane to create two internal cells. And then, excuse me, a septum forms. And the septum is going to be basically separation of the cell wall. All right, and then when the septum is complete, the cells are considered divided. All right, and so here you have two identical cells. Sometimes this is referred to as the daughter cells, whereas this guy up here is the parent cell. And then almost immediately, as long as the conditions are favorable, then these two daughter cells will then start replicating and then be the parent cell. And then you have, this next row down here, you'll have two cells coming off here, two cells here. And that's what binary fission is, and how cell numbers can increase very quickly because you go from 1 to 2 to 4 to 8. And when the generation time, the time that it takes to complete a generation, all right, so basically the replication time, also used interchangeably for generation time. If the generation time is only 20 to 30 minutes, then starting with one cell and after just 24 hours, you have a lot of cells. And generally, when you have an infection or when you get inoculated with an organism, it's just not one organism. Maybe it's 1,000 or 1,000,000. It just simply depends. It's kind of a gross thought, but that's the case. So that's binary fission. Bi is two. They just kind of fuse apart. And generation time. All right. So talked about this already. All right, generation time or doubling time, replication time, the time required for a complete fission cycle from parent cell to two daughter cells. So, as long as the environment remains favorable, all right, the nutrients are there, the moisture is there, the temperature is correct, the pH is correct or within a good range, and there aren't very many toxins. All right, the doubling will continue to go on at a constant rate. Now, this figure, I think I mentioned maybe in section 1, we were looking at the objectives. This is one of my favorite figures in microbiology. All right, this is the growth curve in a bacteria culture. All right, so you have 4 main stages of growth in the bacteria culture. And on the y-axis here, yes, y-axis, you have the number of viable cells, and on the x-axis you have time. And here is your tube, and you have this tan color and very few live cells. All right, and what's going on here in these first 5 hours, if I can find my mouse here. I hope you're not seeing it and it's just being really crazy for you. All right, here's the lag phase, and you see the number of viable cells is remaining relatively constant. All right, it's so small that you don't see any changes, although there may be some few changes in the cell numbers. All right, but this is called lag phase because the number of cells is just lagging at this time. And what those cells are doing is getting metabolically active, replicating its DNA, making sure all the proteins are there that it needs to replicate and divide itself. And then after those few hours, then you have this exponential growth phase, sometimes referred to as log phase. So you have this logarithmic value on the y-axis. Right? It's exponential growth phase or log phase growth. And you see you go from very few cells here to many cells, this rusty Texas orange-like color. Right, so here conditions are favorable, lots of nutrients, no inhibition, and so the growth is continuous. All right, then at this point, maybe 16 hours, and this is going to be varied depending on how much bacteria was there initially, what the culture conditions are, etc. But then you eventually reach the stationary phase. And it's in the stationary phase where bacteria will actually start to die. Why do they die? Well, the nutrients are running out. And as these cells are growing and dividing, there is metabolic waste. And so that waste is being spewed out into the liquid. And so now there's toxins building up. And so you start to see this small amount of dead cells. And basically what's occurring are cells are dying and reproducing at the same rate. All right, so equal death, equal growth. As some cells die, they open up and they can release more nutrients. And so then some of those cells that are still viable can utilize those nutrients and grow a little bit more. And so you have the even line here. Now, after more time, the environment is just very unhealthy. And so at this point, you have more death. Then you have growth, and so you have this negative or downward curve. Now, are there any live cells? Yes. Left? Yes. You can see that by the orange color here versus the gray or black. There actually can be some cells that are going to replicate because, again, as those other cells will die off, maybe some of the hardier, healthier live cells will utilize those nutrients that are being released and can push through all the toxins that are in the environment. But eventually this will come to an end. But if you were to take a small amount of this culture and add it to a very fresh culture like this, you would probably see the same scenario. All right, give it some time, and you take those cells that are alive but not really doing a whole lot because life isn't good, and then they'll take off again. All right, so that is the growth curve. All right, lag phase, exponential or log growth phase, stationary phase, equal amounts of death to equal amounts of live cells being produced. Death phase, more cells dying than are dividing. All right, then concept check. Put the steps of bacteria growth in the correct order. All right, and you have your options of death phase, lag phase, exponential or log phase, and then stationary phase. All right, and if you remember lag, the cells are just lagging behind in their activity. So that's what's going to start it off is lag phase. Then you have that high amount of exponential growth. Stationary phase where you have equal death versus equal life. And then death phase where you have more cells dying off. And that is the end of section 6.3.